Medical nanotechnology, enhancing drug delivery in ophthalmology. Introduction. In recent years, the field of medical nanotechnology has revolutionized the way we approach drug delivery. By harnessing the unique properties of nanoparticles, researchers have developed innovative strategies to improve treatment outcomes in various diseases, including ocular disorders. This script aims to provide a comprehensive overview of the advancements in medical nanotechnology for ocular drug delivery and their potential applications. With a focus on topics such as polymeric micelles, surface modifications of nanoparticles, liposomes, solid lipid nanoparticles, stimuli-responsive nanocarriers, gene therapy, gold nanoparticles, silica nanoparticles, pharmacokinetics, blood-retinal barrier, ocular toxicity profiling, and smart contact lenses, we'll explore how these technologies can enhance drug delivery in ophthalmology. Polymeric micelles hold immense promise as carriers for drug delivery due to their unique properties. These nanosized structures are formed from amphiphilic block copolymers arranged in a core shell structure. Their hydrophobic core is capable of encapsulating a variety of poorly water-soluble drugs, while the hydrophilic shell provides stability and improves biocompatibility. Polymeric micelles have been investigated extensively for ocular delivery, with studies focusing on their potential in treating diseases such as glaucoma and macular degeneration. The utilization of polymeric micelles in ocular drug delivery offers advantages such as enhanced solubility, prolonged drug release, and improved therapeutic efficacy. The cornea represents a major barrier to drug entry into the eye due to its unique anatomical and physiological characteristics. Overcoming this barrier is crucial for effective ocular drug delivery. Surface modifications of nanoparticles offer a promising approach to enhance corneal penetration. By adjusting the physicochemical properties of nanoparticles, such as size, charge, and surface chemistry, improved drug delivery to the ocular tissues can be achieved. Techniques like PE gelation, coating with mucoadhesive polymers, and surface modification with cell-penetrating peptides have demonstrated remarkable results in enhancing corneal penetration. These strategies hold great potential in delivering therapeutics to the anterior segment of the eye. Liposomes, spherical vesicles composed of lipid bilayers, have gained considerable attention as nanocarriers for ocular drug delivery. Their ability to encapsulate both hydrophobic and hydrophilic drugs, along with their biocompatibility, makes them an ideal choice for sustained drug release in the eye. Liposomes can be tailored to modify their release kinetics and improve stability, allowing for prolonged drug retention and reduced dosing frequency. Additionally, surface modification of liposomes using ligands or antibodies can enable targeted delivery to specific ocular tissues. Liposome-based nanocarriers offer a versatile platform for the delivery of various therapeutics, including anti-inflammatory agents, antibiotics, and anti-vascular endothelial growth factor, anti-VEGF, agents. The treatment of retinal diseases often requires the delivery of hydrophobic drugs to the retina. Solid lipid nanoparticles, SLNs, composed of biocompatible lipids, have emerged as an effective carrier system for hydrophobic drugs in ocular drug delivery. SLNs offer advantages such as increased solubility, improved stability, and prolonged drug release. Their small size and unique lipid matrix enable efficient drug transport across ocular barriers, facilitating drug accumulation in the retina. By encapsulating drugs within SLNs, it becomes possible to achieve sustained release and localized targeting, minimizing side effects and optimizing therapeutic outcomes. Stimuli-responsive nanocarriers have garnered significant interest due to their ability to release drugs under specific physiological conditions, such as changes in pH, temperature, or enzymatic activity. By taking advantage of these stimuli, precise control over drug release in the eye can be achieved. Various stimuli-responsive nanomaterials, including hydrogels, polymer-based systems, and inorganic nanoparticles, have been explored for ocular drug delivery. These nanocarriers offer the potential for on-demand drug release, personalized medicine, and improved patient compliance. Gene therapy holds great promise for the treatment of various ocular diseases, including inherited retinal disorders and neovascular disorders. Nanoparticles have emerged as effective carriers for delivering therapeutic genes to target cells in the eye. By encapsulating DNA or RNA constructs within nanoparticles, protection against degradation and improved cellular uptake can be achieved. Nanoparticles can be designed to facilitate gene delivery specifically to ocular tissues, ensuring efficient and targeted therapy. The ability to regulate gene expression and potentially correct genetic mutations makes nanoparticles a valuable tool in ocular gene therapy. Retinoblastoma, a malignant eye tumor affecting children, often requires localized and targeted drug delivery to achieve effective treatment outcomes. Gold nanoparticles, OMPs, have remarkable potential as carriers for delivering therapeutic agents to retinoblastoma tumors. OMPs can be functionalized with targeting ligands, such as antibodies or peptides, to specifically bind to tumor cells, enhancing drug delivery and minimizing off-target effects. Additionally, the unique optical properties of OMPs allow for imaging and photothermal therapy applications. The use of OMPs in retinoblastoma treatment represents an exciting area of research with the potential for significant clinical impact. The delivery of protein-based therapeutics to the eye is challenging due to the instability and degradation of proteins within the ocular environment. Silica nanoparticles have emerged as promising carriers for ocular protein delivery. By entrapping proteins within a silica matrix, protein stability is improved, and controlled release can be achieved. Silica nanoparticles can be further modified to enhance mucoadhesion, prolong retention time, and facilitate penetration into ocular tissues. This approach offers the potential to overcome barriers associated with protein-based ocular therapies and improve therapeutic efficacy. Understanding the pharmacokinetics and biodistribution of nanoparticles in ocular tissues is crucial for effective drug delivery. By characterizing parameters such as nanoparticle size, surface charge, and formulation, researchers can optimize drug release and distribution to target tissues. Different routes of administration, such as topical, intravitreal, or systemic, may influence the kinetics and biodistribution of nanoparticles. Techniques such as fluorescence imaging and electron microscopy enable researchers to visualize and quantify nanoparticle distribution in ocular tissues, providing valuable insights for further improvements in drug delivery strategies. The blood-retinal barrier, BRB, poses a significant challenge in ocular drug delivery, as it limits the passage of drugs from systemic circulation to the retina. Nanoscale drug delivery systems offer opportunities to overcome this barrier and enhance drug delivery to the retina. By exploiting the size-dependent transport properties of nanoparticles, researchers can achieve efficient drug accumulation in the retina while minimizing systemic exposure. Strategies such as receptor-mediated transcytosis, carrier-mediated transport, and disruption of the BRB using permeation enhancers have shown promise in overcoming the challenges posed by the BRB. Safety assessment of nanoparticles is a crucial aspect of ocular drug delivery. Understanding ocular toxicity and potential side effects is essential for the successful translation of nanomedicine to the clinic. Ocular toxicity profiling involves evaluating parameters such as cytotoxicity, inflammatory response, and tissue damage caused by nanoparticles. In vitro and in vivo models are employed to assess nanoparticle toxicity, allowing researchers to identify biocompatible formulations and optimize drug delivery systems. Comprehensive ocular toxicity profiling ensures the development of safe and effective nanocarriers for ocular drug delivery. Smart contact lenses represent an exciting technological advancement in ocular drug delivery. By incorporating nanotechnology, these lenses can serve as platforms for controlled and localized drug release. Responsive materials embedded within the lenses can be triggered by external stimuli, such as changes in pH or temperature, to release drugs in a controlled manner. Smart contact lenses offer several advantages, including prolonged drug delivery, reduced dosing frequency, and improved patient compliance. These technological marvels have the potential to revolutionize the treatment of ocular diseases, providing personalized and on-demand drug delivery. Medical nanotechnology has transformed the landscape of ocular drug delivery, offering innovative solutions to overcome various challenges. The topics discussed in this script provide a comprehensive overview of the advancements in medical nanotechnology for ocular drug delivery. From polymeric micelles and liposomes to gene therapy and smart contact lenses, researchers continue to push the boundaries of what is possible in improving treatment outcomes for ocular diseases. As we progress toward personalized medicine and targeted therapies, the field of medical nanotechnology holds great promise in revolutionizing the way we deliver drugs to the eye. With further research and development, the potential for improving patient care and enhancing quality of life in ocular medicine is vast.